So welcome everyone to our community conversations hosted by today MTM-CNM Family Connection. And we're so happy to invite and welcome Astellas Gene Therapies, who will share an update on the AT132 Gene Therapy for X-Linked Myotubular Myopathy. And we're so excited to see so many families that we have already connections with, the members of the community, broader community, but we also have many new families and new contacts joining us. So we're just going to take a moment to introduce our organization. So our nonprofit, MTM-CNM Family Connection, our mission is connecting families affected by myotubular and centronuclear myopathies to research, resources, and relationships. I am Erin Ward, and I'm here today with Mark Ward, my husband. We had a son who lived 20 years, William, with myotubular myopathy, and we continue on our work with this organization in his legacy and his memory. And also we're joined with our co-founder and partner in all things with organization, Marie Wood. Marie and David have 5 beautiful boys. 2, Reed and Paley, live also with myotubular myopathy, and together we're so proud and feel very blessed to have an opportunity to serve the greater community. Our nonprofit was developed out of grassroots organization efforts to be able to bring together patients, families, industry, academic researchers, and medical professionals over the years at our conferences. And to date, we've held 6 national conferences. And as you can see, it's an incredibly powerful opportunity to bring families together along with the medical professionals in the community to really grow and strengthen our efforts to find treatments and cures and to support one another in this incredible life that our individuals live with every day, facing these very ultra rare diseases. And through our national conferences, we really realized the power of engaging the community, and maybe sometimes without even identifying exactly what that meant. For a lot of the time over the last many years, we've been building clinical trial readiness. And we were very proud of the model that we developed with our national conferences, that were really a collaborative model of bringing patients and professionals together over a course of an entire weekend to be able to really learn with bidirectional opportunities to share stories from both perspectives, and also to accelerate research through offering opportunities to enroll in natural history studies as well as observational studies right on site. Early on, we partnered with industry members who oftentimes were maybe introducing their company to the community and being able to have dialogues about what's most meaningful and most important in therapies to be developed. Additionally, we offer educational webinars and virtual connections such as this, as well as social media, discussion groups, newsletter, blog. But please visit our website for more information. Sign up for our newsletters if you're not already. The last few years, especially through COVID, we've been focusing a lot of efforts on patient engagement and drug and therapy development, and along with the FDA. And together we partnered with Will Cure, another organization in our community. And in 2020, we hosted a patient listening session, as well as a patient-focused drug development meeting. This is a really great opportunity, if you haven't viewed the patient-focused drug development meeting, to learn more about our rare disease community and to hear about what's most important for those of us living with this disease. So I invite you to visit our website and to watch that recording if you haven't already. And part of this patient engagement is having ongoing conversations with our industry partners. And here, that's what brings us here together today. We're so excited to have Astellas Gene Therapies to provide an update. Just really quickly before we get started, you had an opportunity to read this at registration. But most importantly, when we're talking about consideration of treatments, it's always important to refer to your doctor and your qualified healthcare provider when you're discussing medical decisions. And without further wait, we'll get to the highlight of our presentation today, which is introducing the Astellas Gene Therapy team to further update. And I'm so happy to hand over to David Palacio Valero, who is the Executive Director of Patient Advocacy Engagement. Welcome, David.
Thank you, Erin, and thank you, Mark and Marie, for the nice welcome and the invitation to be part of this exciting webinar. We are very committed to share as much as we can with the audience, a lot of expectations regarding what is happening with the program, and also some considerations for the future. So with me, I had a strong colleagues with a lot of experience in the field. So I'm going to hand over to Ha and Christine for them to introduce themselves, and we continue. Ha?
Thank you, David. So hello, everyone. Happy Saturday. My name is Ha Tran, and I am Executive Medical Director at Astellas Pharma Global Development, and I'm currently overseeing the gene therapy programs here at Astellas. By background, I am a board-certified pediatrician specializing in pediatric nephrology, and I've been working in pediatric and rare diseases drug development for nearly a decade now. And I continue to teach and teach pediatric research and occasionally practice out in Palo Alto, California. So thank you so much for having me. And with that, I'll pass it along to Chris.
Thank you, Dr. Tran. So as indicated, my name is Christine Skolas. I'd also like to echo David and say thank you for the invite. I'm grateful to be here today and to share some information. I've been in regulatory affairs for 15 years and just wanted to maybe give a little background on what regulatory affairs is. It's not necessarily a career that in high school you say, I want to be a regulatory strategy person. So what this division is within a pharmaceutical company is we're the direct contact for regulatory agencies, and we're responsible to liaison between our company and the agency. We're the group that submits information to them. We're the group that interprets pharmaceutical law and as well guidances and helps to build a strategy for the development program and also to maintain compliance for any approved drug. So again, thank you for the invite today. Looking forward to sharing information with you.
Thank you, Christine. So now my name is David Palacios Valero. I'm a physician by training. I've been in the industry for more than 20 years and focused in patient advocacy primarily in the last 15, 18. My experience with Astellas started last year in August when I just decided to explore the fascinating world of gene therapies. And I joined the team and also joined forces with people that you might remember, Alyssa Galloway, who just left the company a few months ago, and Hilary Dana, who has been working with us to continue the program in the activities that we have for patient advocacy and engagement. It is really fascinating what is happening in this space. And also it's really fascinating to explore how we can overcome some of the challenges that we all face to really achieve our common goal, which is bringing those valuable solutions for the patients and the communities. With that, next slide, please. You can see what we are doing in the space of myotubular myopathy is started years ago with the dentists. And I'm going to explain a little bit of the trajectory of that. But next slide. And these are disclaimers, so you have the chance to read it, but also just to clarify that this is just investigational. We are not here to promote anything. And it's important to recognize the framework that we are embarking in from the regulatory perspective. Now having Christine on board, we are safe here. Next one. So one of the elements that we recognize in the community is the courage to take a decision to participate in clinical development. Many of you have been making that decision. And we want to take this moment to honor some of the patients and boys who pass away during the process in this Aspire trial. The courage of the families, the support that they bring is just to elevate the ability for us to learn and move forward and to benefit the broader community and in the development of those solutions. So we thank you and your family and your kids for being part of this process. It's not an easy one and it's a long process. So let's see a little bit of what we're going to share with you today to update you on where we are. Next slide, please. So I'm going to give you a little bit of background on what had happened because many of you have been familiar with Astellas now, but most of you have been more familiar with Audentes. It's important to clarify where we are with that transition. The next topic is probably the hot topic here today, which is about where are we, where's the program going, what are the findings today, some of the things that we can share about the whole of the program. And finally, we try to see what is the future, how we can continue this process moving forward. Next slide. Okay, so this is pretty much the transition started in 2021 when Astellas acquired Audentes and became AGT, Astellas Gene Therapies. That is, it's a continuation of our commitment to move the programs forward and bring the value to patients affected with rare diseases in a bigger scale. The ability for us to overcome challenges and complexities, join forces among the experiences that came from Audentes and the experience and the capacities that Astellas can bring to the space is amazing. Many of you probably heard about the investment, the huge investment done by Astellas in the manufacturing plant in North Carolina. This is one of the signals of our commitment long-term with the space of gene therapies, and particularly in the space of this particular program. Next slide. So our mission continues and remains in terms of find some transformational elements. And again, regulators are not happy to just see minor improvements. They want to see something transformational. And to achieve that, we count on you, we count on the families and the patients that are part of the trials. And we stand here to combine those benefits between the experience that we had from those who started the program and the future expectation with the people and the talent that you are going to see today, and probably more people in the future. We are very excited to be able to reconnect with the community. I think is one of the points that we missed a little bit during the last year and a half or two for different reasons. But it's exciting to have to reconnect and continue the learnings and the learning from what we achieved, what we found, and be able to share with the regulators what we found, but more importantly, keep you posted about what are the conditions to continue our trajectory to find the solution for the MTM patient community. With that, I'm gonna, let's say, hand over to Christine, that she will continue giving more details about what is happening with the whole of the program and details about the evolution of these steps. Thank you.
Thank you, David. So as many of you know, that our program for AT132 for the treatment of XLMTM was placed on clinical hold in September of 2021. So what does clinical hold mean? Clinical hold is an order imposed by the FDA to suspend the investigation. So the study that was suspended was named Aspiro. And so if you go on clinicaltrials.gov, you'll see that information. So what does this mean when we're put on a clinical hold? No additional participants can be recruited, screened, or dosed or enrolled into the Aspiro trial. What happens when why do companies get placed on clinical hold? There could be several different reasons. The number one big reason is for safety issues, and that is the case for us as well. So in September of last year, we notified the FDA when the last patient that was dosed was experiencing liver issues. The FDA and the company put ourselves on a temporary pause of the investigation. FDA called us immediately after obtaining the information that we shared with them about the safety issue, and then they verbally put us on clinical hold. 30 days later, after evaluating the information that we've submitted to them related to that safety issue, they issue us a letter, a formal letter of clinical hold. And in that letter, they express concerns that we need to address in order to have them evaluate if the clinical hold can be lifted. So in the interim time between receiving that hold and communicating a response back to the FDA, the company then does investigations to investigate what could have been the problem that caused this safety event. And as you can see in the middle of this slide is kind of a step ladder approach to what I'm describing. So right now we're in that investigation phase for the investigating what it could be about our product or this trial that led to that safety event. Sometimes resolutions will be modification of the protocol. Sometimes it's that you might need to end that study and start a new study with different elements to ensure a benefit risk profile that's more favorable to the safety and efficacy under evaluation. Sometimes it's enhancing the drug product that's under investigation. Might be something that needs to be tweaked with that product. So there's a lot of different things that can happen over the course of this time between that letter being issued and resolving what the problem is to get back into the clinical trial in a more, so that we're ensuring a more safe environment or profile for those patients. So after we've gathered this information, come to a resolution, what we do is we'll draft a response to the FDA, submit it to them formally. They have 30 days to review our response and determine if we've satisfactorily answered their questions and that they agree that it's safe for us to return to the clinic. There's still some steps that occur after we receive that letter back from the FDA, because the sites themselves also have to agree that those changes are enabling for improved safety. So as you know, investigational review boards at the site, they as well will take a look at what has been changed, and then we'll need their agreement as well that yes, it's safe for you to return to clinic and for patients to be dosed. This is a global program, so in addition, any other countries that we have a clinical trial in, the study has been temporarily halted in those countries as well. So we have to take similar steps in those additional countries where we have sites. So I hope that was informative for all of you. We're still in the investigation phase, but I would like to transfer the conversation over to Dr. Ha Tran, who can give you a little bit more details about our study.
Thank you, Christine, for the informative information. I hope that was helpful for the community. So let's move to the next slide. So thank you. So this slide is intended to explain on a very high level how AT132 intends to treat XLMTM. So as you are all aware, XLMTM is a monogenic disease, meaning that it is a mutation on a single gene due to changes on that single gene MTM1, which encodes myotubularin. Now, myotubularin is a protein required for muscle cell differentiation. And so if you look on the graphic on the top right there, you see with a healthy muscle. So the four large hexagons are meant to represent the myofiber or muscle fibers. And each of the circles inside represent myofilaments. Inside myofilaments contains actin and myosin. And so those are the contractile proteins, and without them, muscle is not able to contract. So rendering, basically, you do not have ideal muscle function. So in the XLMTM muscle, you can see that there are reduced myofilament density. So it's much less filled in than on the other side, pushing the nuclei towards the middle versus on the periphery. In terms of AT132 itself, below right corner is a schematic of the vector genome. And so there are the pieces are the RAAV8, that's recombinant AAV8, which is the viral vector. And we have Desmond as the promoter, and then the humanized MTM1. This construct is delivered in an IV formulation as a single dose, where it's expected to enable the production of myotubularin for the participant's lifetime. Next slide, please. So this is a slide that demonstrates the respiratory function in the participants through week 48. And just kind of in summary of the Aspiro study, so a total of 24 XLMTM patients were dosed in the Aspero study at 2 dose levels. So the lower dose level you see in blue was at a dose of 1.3 10 to the 14th vector genome per kilogram. And then we had 7 patients dosed at that level. And then the higher dose level was 3.5 times 10 to the 14th vector genome per kilogram. We did have 4 deaths in total, 3 of them occurring at the 3.5 e to the 14th dose at the high dose, and then one death occurring on the low dose. So this figure is intended to demonstrate the various respiratory ventilation findings that we had. So the top line, I wish I had a cursor here, but the control is the gray, thank you, is the control group. And the control group, we had 14 patients that were a part of Inceptus and also a part of Aspero who were in the control group. Their baseline hours of ventilation was approximately 19.9 hours, so approximately 20 hours. And by week 48, as you can see, throughout the duration of those 48 weeks, the ventilation time was pretty stable, right? So no real changes. And at the end of week 48, it went up slightly to 21.3. In comparison with the low dose group of 1.3 times 10 to the 14, the blue line, you can see that the baseline was close. Baseline ventilation hours on ventilation was approximately 21 hours. And through the course of the study, that had decreased. And by week 48, it was just about 1.3 hours per day. In comparison, the dark red line was the high dose group, also started at a high ventilatory time at approximately 23.6 hours. And then over time also had decrease in hours of ventilation per day. And then by week 48, it had decreased down to 9.7 hours per day. And then as of our data cut of February 28th, 2022, all of the data that I present today will be based on that data cut of approximately a year ago. So currently among the 20 surviving participants, 6 out of 6, so all of the surviving participants in the lower dose group, and 10 out of 14 in the higher dose participants have achieved ventilator independence, meaning that they are no longer on the ventilator. There is one participant in the lower dose group who continues to require intermittent non-invasive ventilator support during illnesses. Next slide. So this slide describes what we found up until last year, which is February 2022. And these go through the motor milestones. So I'll start with the right panel there, the right figure. So the right figure are the control group. So those are the 14 patients. And then if we can just go through the key, just so that everyone is able to understand. So the black child, the black dot represents one participant who is able to sit unassisted at baseline. So that's participant 30, you can see, who is able to participate, already sit at baseline. And then the blue demonstrates the ability to sit without support for more than 30 seconds. So in the control group, there were 1, 2, 3, 4 boys who were able to sit without support for approximately 36 seconds. However, none of the participants in the control group were able to rise to stand alone, walk without support, or walk out stairs. Now, in contrast, when you look on the left side, so in the top figure there, you can see that there was 1 out of 6 patients who did sit on his own prior to being treated. Subsequently, without being treated, was able to stand. And then of all the surviving participants, all of them were able to sit by the end of our data cutoff. And then additionally, you can see that the milestones were achieved with walking without support as well. Okay, and so in the higher dose group, you could see that there were also participants who were able to sit unassisted as well for at least 30 seconds. Okay, next slide. Okay, so now I just want to give an overview of the 4 patient deaths and what we've learned thus far. So as Chris mentioned earlier, that the study is currently on clinical hold. And in 2020, 3 of the patients who were dosed in the higher dose level died. And then the study went on clinical hold at that time. Audentes or Astellas Gene Therapy at that time made some changes to the protocol to ensure that there would be additional monitoring, and made some changes to the inclusion and exclusion criteria. The decision was made to go into to resume the trial, and had the FDA's okay at that time to go in at the lower dose. And unfortunately, in September 2021, that additional participant who was dosed at the lower dose level did have the hepatobiliary serious adverse event, which progressed to fulminant liver failure and eventually died. At that time, when the company noticed that the participant was starting to have those liver changes, had voluntarily placed themselves, pause enrollment of new participants. The FDA did place the study on clinical hold, and we have been on hold since then. When we look at these events, we see that all 4 study participants, as well as more than half of the subjects in Aspiro, had some sort of evidence of a pre-existing hepatobiliary disease, meaning some abnormality in the lab values of the gallbladder or the liver. However, at the time of enrollment, they all met inclusion criteria, meaning that they didn't have any signs of clinically significant underlying liver disease at the time of enrollment, and their liver enzyme values were not greater than 5 times their upper limit normal, which is by definition abnormal. Additionally, there were no signs or symptoms of hepatic pileosis, which is a rare condition with blood-filled cysts in the liver that can lead to liver failure and liver bleeds. This condition has been associated with XLMTM patients. So as Chris mentioned earlier, the investigations are ongoing, but the results to date do not suggest that these events were immune mediated. Oftentimes with AAV associated gene therapies, there are concerns for immune reactions against the vector or the capsids or the transgene. And to date, our data does not suggest that this is something that had occurred. Next slide. Okay, so in terms of the status of the investigation into these deaths, so we've been working to understand the mechanism of cholestasis in XLMTM and how this could be exacerbated by AAV gene therapy. So these types of activities have consisted of looking at the autopsy reports, reviewing the findings that we saw, and then also there have been some animal and in vivo studies to look at the liver cells. What has been found was that there is a bile salt export pump that has a major mechanism for bile acids export from hepatocytes, and it seemed to be it was found to be missing in the 4 boys who died. And so this mutation is similar to one that we've seen with another type of liver condition. So the ongoing hypothesis is that there is a role or somehow an interaction with AT132 that could have led to an absence of BSEP, or whether or not that's something that is common in XLMTM patients. So we are seeking to understand whether or not this could have contributed to the depletion of BSEP in these individuals. And with the additional information that we find, we plan to modify participant eligibility criteria to reduce risk of hepatic toxicities. Additionally, the company has been working to enrich our product in line with industry trends to constantly improve our product and perhaps remove further empty capsids from our investigational product. We have been continuing to engage with our external experts, many of which whom you know, and with our patient community as well. We have ongoing discussions with our regulatory authorities to address key items for the clinical hold, and to ensure that, should we be able to come back to the clinic, it would be in an area where, we are benefiting the patients who are at their lowest risk of developing any serious adverse events. So, we have had interaction with the FDA in the beginning of November 2022. And, we'll also have a meeting with them actually on Tuesday to discuss some of our CMC topics. We've also have been meeting with the European medicine agencies back in the end of November. And, we have ongoing plans to discuss, and, when the time is appropriate, we'd love to come back and share more information. Next slide. So I'll hand it to David.
Yeah, thank you very much. And again, thank you for the audience for the patients for a bunch of information, acronyms, and technicalities that are easy to follow. That's many of you have been more expert than us in that context, but the messages are really that.
Hey, David.
Yeah.
I'm sorry to interrupt. I don't know if it's just me, but I think we're having some challenges with your audio.
Oh.
Sounding a little scratchy.
Headphones right now.
Yeah, I think we'll give you a minute to maybe see if you can reconnect your audio, and I'll just jump in briefly. So as we're coming to the questions section here, again, if anybody attending has questions based on the presentation we've just gotten. So first of all, thank you very much to David and Chris and Ha for sharing this information. We do have some time for questions. Please type those in the Q&A as well. And I'll see if David maybe has his sound back. Maybe not quite yet. So while we're waiting for him to reconnect, maybe I'll just direct a question to Haas. It's sort of on one of your last slides there. It's a little more on the technical side, but you mentioned the biosalt export pump. Can you just give a little bit more description of what that is and how it might potentially be important to liver function and for somebody with MTM? I know it's an ongoing area of investigation, but it's something that hasn't really been discussed in the MTM community till now.
Correct. Yeah, so it's still a hypothesis, and a lot of it is based on the findings that were on the histological reports from the autopsy, which were performed by Dr. Mike Loyler, and I know he has been presenting it and plans to publish it as well. But there is, when we did ask Dr. Loyler what is different about the autopsy reports? And is this something that, you would see with other gene therapy programs? And the answer is no. What we find is something very unique. And right now, we're trying to determine whether or not the myotubulin deficiency may somehow contribute to this absence of this bile salt export pump. So this is something that's needed to export the bile out of the gallbladder, right? And so how these patients presented was that they would have this backup of bile in their gallbladder, and once you can't excrete it, it leads to liver injury. And so this is something that is ongoing. In terms of the critical path for development of a drug, maybe I could pass to Christine.
Yeah, happy to help contribute to that answer. So FDA they are aware of the criticality of getting treatments to patients in the rare disease space. There's several guidances that the FDA has published in this regard. For the treatment of rare diseases, for use of gene therapies for rare diseases. So that's something that sponsors can look at and utilize. The other thing the FDA has done is they have different programs to help accelerate the investigation and approval of these conditions, because number one, they're rare diseases. Sometimes as indicated, they have high morbidity or mortality and low quality of life. So the FDA is very conscientious of this. However, the FDA also requires that we follow the law, and they have to have a well-designed study so that the data they review is not biased, so that they can approve that investigational product, knowing that they're going to ensure safety for the patient, and that it's going to ensure that those patients have some form of efficacy as well from that drug. So it's a balance between making sure that you're designing your trial so that the data that you get out of it is not biased in any way, and also making sure that you can apply to their programs that they've put in place to try to speed up the ability to conduct your study, as well as for them to review your study. A lot of times in this rare disease space, you don't need two well-controlled studies like you would see maybe in a drug that's going to treat hypertension or high blood pressure. In those cases, you see hundreds of patients that have been evaluated, and there's two well-controlled clinical trials. But in rare disease spaces, generally you can truncate those studies into one or two instead, and then you have less patients, because in the case of a rare disease, you don't have high numbers of patients to perform this investigational study in. But it can be a tricky balance because the FDA and EMEA do need data that they can assess and make sure that they're providing safety and efficacy to the public or to the children or to the adults with these rare diseases. So I hope that helps somewhat with this answer. Happy to, if there's a follow-on question, happy to answer that.
Maybe if I can just add a follow-on to that. Regarding safety and efficacy, right? Sort of the two sides of the coin there. Ideally, everything would be perfectly safe and 100% efficacious. Reality is no drug or no therapy is either of those. For a rare disease and a particularly severe one, such as X-linked MTM, how does Astellas and how does the regulatory agencies, whether that's FDA or EMA, how do they balance the trade-off? I know from the patient community, we tried to convey our voice directly to the FDA through the PFDD that Aaron mentioned, but oftentimes we're faced with a certain clinical course that we all understand very well, and it's extremely challenging, to say the least, right, from just a medical and daily life. So how does that get weighed in terms of either approving or proceeding forward with additional clinical studies that we know are not 100% safe or efficacious?
That's a really good question. And FDA does listen to the patient voice, and they're very concerned that you're picking a primary endpoint that's clinically meaningful to the patient. So in the case of XLMTM, it's been widely agreed from the patient, my understanding, from the patient groups, from the agency, from even Astellas' point of view, that reduction in ventilator is a significant clinically meaningful benefit to the patient. Now, you saw in some of the slides that some of these boys, but not all of them, are able to stand independently. Some of them can even walk. That's a great improvement for some of those boys. But if we have that as an endpoint that the FDA has to measure against, that's a really high bar, right? And so how it's a balance then to understand what's clinically meaningful for the patients, the families, the caregivers, the FDA, as well, and a target that the sponsor can measure in a non-biased way so that it can be assessed whether this is an improvement that's happening spontaneously or if it's an improvement that's happening because of the drug that you've given the patient. For safety, that's a very good question. And it can be really tricky too, because before you even get into your first in human study, you need to prove that your investigational product has been safe in animal models. In our case, we didn't see any cholestasis or liver problems in our mice, in our dog studies, and in the non-human primates. But you can get surprised when you put that drug into a human, because again, we're not like for like, right, for all those animal models. So obviously death is highly concerning to the agencies, and they take that very, very seriously. And so that's part of the reason why we're having intense investigations that we're conducting and continuing dialogue with the agencies to resolve this. And maybe how you want to add some more of your context for the safety?
Yeah, absolutely. Thanks, Christine. You know, the FDA had held a OTAT town hall. I believe it was just this past Monday, so I'm not sure if anyone from the community was able to attend. But they made a really good point in terms of, you know, if they are aware of the risk with gene therapies, but they're also aware of the potential transformational benefit as well. And for them, one key point they mentioned is that, you know, the patient selection, like we need to ensure that the patients, the selected, are healthy enough, you know, in the context of having this severe disease, that they are healthy enough so that if they do encounter an adverse event, that they could, you know, recover from it. And so, you know, from our clinical trial, from the Aspiro data, it was remarkable that, you know, even there were patients who had serious adverse events of the liver and did recover, and then they were able to achieve those benefits, right, coming off the ventilator and meeting some of those milestones, which are so important. So all of these things are how we determine an acceptable risk benefit, is really just working through that and really selecting the right patient population.
If I may add, and sorry for the clinician back now, the FDA is, as somebody mentioned, open to listen. So the question then would be how to accelerate the uptaking of the information that they're gathering from the patient community in a way that is translated into a process that is acceptable for all. So sometimes there's a process in place that doesn't match with the current trends in technology and science. So we're learning along the way. That's part of the big challenge for all of us. But also that's why we are here with you. We are working with you and working with others together, because it's not a simple task to overcome. So we will continue open the dialogue, connecting the dots, and try to find out ways for bringing this that they call risk tolerance balance, where they can accept that the community might be taking some risks based on the benefits that they can gain from the gene therapies in general.
Thank you. I'd like to shift to a slightly different thread here, and understanding you probably can't talk about, you know, specific timing for approval, but let's talk a little bit more about this process of what does it take and what can we as a community expect in terms of potentially lifting the clinical hold and hopefully at some point resuming the trial. So first of all, Christine, I think you had mentioned, once the clinical hold is lifted, which again is something we hope for, but we don't know when that may or may not happen. But once it is lifted, there are still additional steps that would need to take place before an additional patient could be dosed. Could you just expand on that a little bit in terms of what that process would be like?
Yeah, happy to do so. So, and I saw a question on here too, so maybe I'll pair it together on when do you, like, is there a deadline to respond to the FDA? So there's really no predefined deadline to respond to the FDA. We are engaging the FDA with formal meetings to share with them what our investigation findings, our proposed path forward to return to the clinic, getting their early feedback on it. But then we also have to provide them formal submissions of our data. And then they'll take that data and review it internally to decide if the data we've generated or if the modifications that we've made to the study are sufficient to return to the clinic. After and in association with those submissions where we give them that data, we'll also respond to the questions that they had in the clinical hold letter. So it's not necessarily just here are your 5 questions, please answer them, you're good to go. There's a lot more involved in this than maybe what it seems on the surface. So let's say that we've submitted all of our data, they decide, the FDA says, great data, we agree with your path forward. Then, and they'll look at the specific questions too. They asked us and they'll indicate, okay, you answered those questions sufficiently. Then they'll issue us a clinical hold release. So after that point, then we have to share the updated protocol with all of the investigational sites. At those hospitals or those sites, they have an investigational review board. And so that review board as well has to look at our modified protocol, our modified informed consent form, our modified investigator brochure, additional document that's involved in clinical trials that describes our study to the physician. And then they'll say, yes, we agree with the FDA, we agree with the sponsor, you can start the study back at our site. If they have a concern, we'll give that concern back to the sponsor, and the sponsor has time to respond to it. If they respond adequately, then the studies can continue. If we don't respond adequately, they may have a suggestion that says, we want you to add this additional modification to your protocol before we agree that you can return to the clinic. So that's kind of the different levels of kind of governance to make sure that when we return to the clinic, that there's different levels of review to make sure that we're ensuring safety when we get back to those, when we return to dosing those kids.
Yeah, thank you. And so there's a number of, I guess, iterations and reviews and approvals that, and some of them certainly with a potential clinical site, I imagine can't even begin until the FDA clinical hold has been resolved, right? Because changes to protocols, for example, and this is one thing I want to get into, for example, age restrictions, right? So we know from the very first iteration of Aspiro, there was an age cutoff. And so there are some questions around that, like what do we anticipate in terms of age? But that might not be defined yet. So you can't have a clinical site review that protocol until it's approved through the FDA. Is that
you
correct?
got it exactly right. Stepwise approach. So after we get the FDA's agreement on our protocol, then we'll share it with the sites. And the sites want to know that too. They'll ask, if they did receive a protocol from the sponsor, they'll ask, did FDA agree to this? So, and then you'd share that with them. Yes, indeed they did. And sometimes you share with them the letter that the FDA gave you. And, sometimes it's just an email confirmation. The other thing, I think there was a question on here regarding age, and in Europe, we do, for drug development, and in the US too, there's commitments to evaluate the pediatric population and to be able to evaluate if you can expand into the adolescent age group if you went for the pediatric initially, or to expand into young adults. So from like going all the way up to age 18 for kids. So regulatory wise, both the FDA and EMEA try to have programs in place so that sponsors are expanding and evaluating how to cover pediatrics all the way up to age 18. That's also a stepwise process. A lot of times you have to, firstly, in an ideal world, you would prove efficacy and safety in an adult, and then you would transfer that investigation into children. But if you, in the case of XLMTM, where you don't have very many adults that have this condition, you would start with the most severe group, as we have, as you've seen. But then you may have to, as Ha had indicated, go with the group of kids that most benefits from this, from a safety and efficacious perspective, and then expand out after you've learned more about your drug product, more about that safest age group, and then take that learnings and apply it to expand it to other age groups later, if you can.
Yeah, and just to speak further to that, I see a question from Chris about the hypothesis of the difference in performance in ventilator independence between the high and low dose group. So just to kind of review Aspiro. So Aspiro was a dose ascending study. So started off with 6 participants who were dosed at the low dose group, and then went on to the high dose group. So the participants in the low dose group were a bit younger and healthier. And that is part of one of the hypotheses where this is possibly why they had it seems to have better outcomes in terms of ventilation and also with the motor milestones as well. I don't know if you want to go back to the slide, but I apologize if I didn't explain it that well also on the motor milestone slides as well. So in the low dose group, so for raise to stand, there was 6 out of 7 patients, which is 86% in the low dose group. The high dose group was 6 out of 17, which is only 35%. And then for walk unsupported, 5 out of 7 in the low dose group, which is 71%. And in the high dose group, 3 out of 17, which is 18%. But compared to the control group, like none of the children in the control group were able to raise to stand or walk unsupported. So that is our hypothesis.
Yeah, so just to underscore that question, I think, or maybe restate it, is it seems counterintuitive to most, and I'm sure to you as well, that typically the higher dose you would see a larger effect, at least in these samples, right?
Mm-hmm. But I think that goes also like when you think about the patient selection as well, right? Which is also something that the FDA, will repeat as well. Is like they really want the sponsors to select the patient population who are at the highest probability to benefit and at the lowest probability to have an adverse event or not recover from an adverse event.
I'll just point out too that during the course of the study, the weaning protocol was changed between the low dose and the high dose group. So that could be a point of variability. And so this is an example of potential bias in the data that the FDA is concerned about. So they want to know that the way you collect the information is the same between groups so that they can adequately compare between groups.
I have a question also just in terms of this type of data that we're looking at. So understanding while the clinical hold is in place and until and unless all those other steps we talked about happen, there will be no additional patients added. But there are a number of patients that were previously dosed. Are they continued to be followed? Is data collected? This data was a cut of almost a year ago. So will there be additional data to be shared at some point in the future just for the existing study participants?
Yes, so data collection is ongoing. So the patients have been, the initial protocol was to follow up through 5 years post-dose. We have amended the protocol and asked for patient consent to follow up for a total of 10 years post-dose. In terms of when additional data will come out, we have the authors of the manuscript have recently resubmitted And so we're waiting for that to come out into the public domain. And at that point, we could share additional information once it's in the public domain. And a lot of the investigators and the experts are also presenting at various congresses, and we work with them. And, once that comes out into the public domain, that could also be shared. In terms of additional data analysis as data cut, we will pair it with the next opportunity that we have. So many of the patients have reached 5 years post dose, and so we will need to look at a good time where we can have a data cut, maybe pair it with a health authority request or an upcoming report that we need to do, so that we could gain additional insights.
I would just add, I think that there's been questions, and we discussed some of the risk benefit decisions and sort of that ethical dilemma, in a sense of we have a severe life-limiting disease that and here's a potential. We certainly have some data on efficacy. We also have some serious safety issues. So I think continuing to evolve that data and get a fuller picture is maybe part of the formula, part of the discussion for weighing that risk benefit in the long term. Right? This data we're seeing here is less than 1 year in length for any given participant. So I think that's something that we also have to understand as a community, is that this is not flipping a switch for any individual, that they're either treated or their symptoms are completely resolved or not, or they're completely safe or not. It's a more complex ongoing picture than that.
Yeah, Mark, I know that we have a sharp stop at 12 and there's a minute left. I just wanted to answer one question about the learning from this period of communication, transparency with the community. We are committed to reopen doors in a proper manner, understanding the limitations, and also have these proper channels to continue this dialogue. There's nothing that we can do without working with you, and this is a commitment that we have. So whatever had happened in the past part of the transition, we learn from there, but we want to move to the next level. But we thank you all for the participation and for the space.
Yeah, I appreciate that comment very much. I know they're been trying to keep an eye on the questions as best I can. There are a number maybe that we didn't quite get to, but hopefully this was helpful for the community. We really appreciate the dialogue and the willingness to be open. It's a challenging topic, right, for a lot of reasons. But we thank the community members for joining, but also David and Chris and Ha for sharing your insights with us. We do hope that this, as you said, David, will be a continued pattern of openness and transparency and dialogue between the community and Astellas. So any final comments or questions from the panelists? And then I'll hand it back to Aaron and Marie.
I just want to really thank everyone for coming and showing your engagement through the many questions. I see many of them did not get answered. So Mark, if you can capture these and take them to us, and we will respond in an email dialogue and return to you, because we are we're just so happy that you're engaged and have a lot of questions, and we'll try to answer them to the best of our ability.
I want to echo the gratitude for the time to spend with you today, and also for the excellent questions we received.
I just have one comment, and then I'm going to hand it over to Marie. But I just want to thank everybody that attended today and helping to continue this important conversation, and especially thank all of the families that participated in the trial to date. Your bravery and courageousness in terms of making those decisions, and we always are continuing this work, especially with those that have passed away in mind. And together we will continue to make improvements and hopefully open doors to many more options within the community. I'm going to turn it over to Marie now.
So thank you again to Ha, David, Christine, and of course Mark and Aaron and everybody who joined. these are pictures from some of our MTM-CNM family conferences. As Aaron mentioned earlier, the connection exists in order to connect families and our allies to resources, research, and relationships that can empower us to thrive in the present and also to achieve our hopes and our goals for the future. So we just really appreciate you guys being so motivated and sharing your voice and your opinions. We're very passionate about motivating collaboration and advocacy at every level, participating in the clinical trials and the development and all of these things, legislative advocacy. There are a lot of different things that we are involved with. And so if you want to connect with us, find out more, be a part of what we're doing, you can follow us on Facebook and Twitter. We have a Facebook discussion group for the MTM-CNM. It's called MTM-CNM Family Conference as well, and our website. So thank you again for joining us, and we hope to hear from you soon. Keep in touch.